So let's continue talking about Chapter 8, Muscular System. Today we're going to focus on the physiology of muscle contraction. We're going to look at the sliding filament theory. Now remember we said that those myofibrils have fibers called actin and myosin. And this picture here we have the actin. Remember the Z line has the actin attached to it. There's another Z line. Remember from Z to Z is a sarcomere. Here's the thick myosin protein filament found in a myofibril. There's the actin, the thin protein filament, and there's the myosin. When sarcomeres shorten, thick and thin filaments slide past one another. So we're going to talk in detail about this, and you need to realize that the actin and the myosin themselves do not shorten, but they slide past each other, thus the sliding filament theory. Okay, the H zone and I bands become more narrow as these slide past one another. This area is going to become more narrow. So here's the H zone, and you can see where these actin filaments overlap, so the H zone is totally missing. The A band is still the same distance, but the I band, the distance between the myofilaments, has become more narrow. The sarcomere, the whole structure, has become shorter as this muscle is contracting. When we look at a muscle slide in the lab, we are looking at a relaxed muscle. It would be difficult to take a specimen of a contracted muscle and see that on a prepared slide. Okay. Now here we have the thick filaments, the myosin, and the thin filaments, actin. The myosin is going to have these structures called cross that protrude off of the surface of the myosin, this thick protein filament. And here's the actin. The actin is composed of a filament, the protein filament, associated with troponin and tropomyosin. Now troponin is this globular protein. Think of this, okay, so this is the protein filament. Think of it as a sessile rope. A sessile rope is sort of wove together and looks like this, and you'll see the individual filaments of that fiber that's found in that rope, and as you untwist it, things begin to happen. So this globular protein troponin is embedded or attached to the tropomyosin. If you had that rope and you wrapped a thread around that rope or a piece of yarn, that would be what the tropomyosin looks like. And the troponin is a globular protein attached to the tropomyosin. So the thick filament, myosin, it has the cross bridges. Okay, the actin is a protein filament, and these areas are where you will have an open site for cross bridging. It has the globular protein troponin, and the tropomyosin is the troponin is connected to the tropomyosin. Now remember the neuromuscular junction, where you have the mitochondria and you have the synaptic vesicles. The synaptic vesicles hold acetylcholine, and acetylcholine is very important. And you have the influx, remember the influx of the calcium here for the synaptic vesicles to open up and release the acetylcholine. It goes across the synaptic cleft to the receptors found on the motor end plate, that folded sarcolemma or cell membrane of the muscle. Okay, right here, motor end plate. And the acetylcholine goes across, and it allows for the influx of calcium and that's very important, and we're going to find that in just a minute. Remember that each muscle each muscle has at least one motor nerve associated with it, and that a nerve is a bundle of nerve fibers. This is the axon, and it is a nerve fiber, motor neuron nerve fiber called an axon, and at the end here is the synaptic knob. So nerve impulse causes the release of acetylcholine. We went through that. That acetylcholine binds to the receptors found on the surface of the sarcolemma in the motor end plate. It generates a muscle impulse. Now, an impulse is also called an action potential. In our little video that the animation you'll watch, you'll see an action potential. An action potential is like a nerve impulse on this nerve fiber. It goes across and it causes the polarity of the membrane to change, and it's actually a bioelectrical impulse that causes things to happen. So the muscle impulse is similar to a nerve impulse. So the nerve impulse happens on this nerve fiber, and then the muscle impulse that happens on the sarcolemma. It is all due to the change of the positive and negative location of ions or chemicals found on the surfaces of the nerve fiber and of the sarcolemma. The muscle impulse eventually reaches the sarcoplasmic reticulum. Now, when the nerve impulse meets the sarcoplasmic reticulum, one of the main things that happen, the most important thing that happens, is calcium ions are released. Calcium ions. Now, you've heard of calcium before. It is an element that is very important for the strength of the bones. But your body, we've already talked about the skeletal system, your body will release hormones when the calcium levels are not high enough, because calcium needs to be plentiful for nerve impulse and for muscle contraction. If you don't have enough calcium, you can't have muscle contraction. Or you can't regulate muscle contraction very well, so you may have tetanic contraction, which is where the contraction is held and not released like a Charlie horse. So that impulse causes the sarcolemma to change its polarity too, like that bioelectrical impulse. And when that impulse rides over the sarcoplasmic reticulum, it causes calcium ions to be released into the cytosol. The calcium ions bind to the troponin and changes its conformation, changes its shape. Conformation means shape. Okay, so here again, here's the troponin, there's the tropomyosin. The calcium ions attach to the troponin. When the calcium attaches to the troponin, it causes the active sites on this actin to be exposed. Now, here they're not exposed, here they are exposed. The calcium ion attaches to the troponin and changes its shape, and it causes, like, remember I said it's like a rope with yarn twisted around it? Well, it sort of untwists this actin protein and exposes these bridging sites or active sites on this actin. If they're not exposed, cross-bridging does not happen, and you will not have muscle contraction. So you have to have enough calcium ions to attach to the troponin to change the shape of the troponin, which changes the shape of the actin molecule, with the tropomyosin exposing the active sites on this actin molecule. Okay, the position of the tropomyosin is altered, the binding sites on the actin is exposed, and this will allow for the cross-bridging to happen, for the myosin heads to touch the actin, and then cross-bridging happens. Now I'm going to go through something real quick and review this with you. Remember when we were talking about a little bit of chemistry at the first of after we talked in chapter 1, we talked a little bit about chemistry, and I told you for molecules to be built you have to have energy. When molecules are broken or compounds are broken down, energy is released. Now the universal energy carrier is ATP. Okay, now that's triphosphate, 3 phosphates. ADP would be diphosphate or 2 phosphates, and you see the second phosphate here. So on these myosin heads you have ADP plus a phosphate. Now what has happened as ATP has come in, it is broken down into ADP and phosphate. So when you break a compound down, what did I say? Energy is released. So that energy is the fuel, or it's like charging a battery. Okay, this has the energy, this myosin head has the energy stored in it. It has to have that energy for this cross-bridging to happen. Okay, so you see the ADP here, ADP here. Now after the cross-bridging happens, you have to have ATP, and it's broken down from ATP to ADP and phosphate, and it recharges these heads again. But I'm getting a little ahead of myself here. Okay, what happens first? You have the actin and myosin cross-bridging. You have to have the cross-bridging. You have to have these active sites exposed, and the only way you can have the active sites exposed is if you have calcium that's bound with troponin. The tropomyosin changes the shape of this actin filament and exposes these active sites. In the cross-bridging binds the actin to the myosin. Then you have a power stroke. Okay, so this is causing those. Remember I said that the actin and the myosin do not change lengths. You have the crossing over of the actin and the myosin. It's sometimes referred to as a power stroke. The myosin cross-bridge pulls the actin along. ADP and phosphate releases from the myosin. Okay, there's the ADP and phosphate. The power stroke happens. In that power stroke, it really doesn't show it here. Well, here it is. The cross bridge pulls the filament along. The ADP and phosphate are released in this step 4. And the releasing of these, this is the power stroke, comes from the energy stored in these heads from the breakdown of ATP to ADP. Okay, these are released, and then ADP and phosphate can be, or ADP can be phosphorylated back to ATP through cellular respiration, and more ATP is needed. ATP will attach to these myosin heads, and when the ATP attaches to the myosin head, then the cross-bridging stops. You see a gap here. Okay? Then the ADP and phosphate, the ATP splits, which provides power to cock the myosin cross bridge heads again so it's ready to go. It's like, okay, you've set up a row of dominoes and now your dominoes have fallen over. You've got to reset everything so you can do it again. You've got to re-energize these myosin heads so a cross bridging can happen. So ADP binds to the myosin and it's released from the actin. The linkage between the actin and myosin cross bridge break when these ADPs attach. The ADP is a larger molecule that splits to a smaller molecule. To build an ATP, this ATP has stored energy. And when you break it down to a smaller molecule of ADP plus the phosphate, you are releasing the energy, and the energy is stored in this head so cross-bridging can happen. The myosin cross-bridge goes back to its original position. As long as you have calcium, as long as you have ATP, you can have more cross-bridging. You have to have the calcium, you have to have the ATP for the cross-bridging to happen. Now, acetylcholine at the neuromuscular junction causes the influx of the calcium that goes across the sarcoplasmic reticulum and the release of calcium. All happens because of acetylcholine. If, as I said before, if acetylcholine stays present and you have calcium and ATP, you can have cross-bridging, but you don't need a continuous muscle contraction. You must have muscle relaxation. If you have calcium and ATP and the presence of acetylcholine, you're going to have continuous muscle contraction. You don't want that. So to stop the muscle from contracting, you need this enzyme that is released on the motor end plate and into the synaptic cleft. Acetylcholine esterase, an enzyme, is released. Now, an enzyme is indicated by an ASE ending of the word. So anytime you see acetylcholine esterase, this ending, it indicates that it is an enzyme. And in this case, this enzyme's job is to break down acetylcholine into acetic acid and choline. It's taken up into the synaptic knob and comes together again as acetylcholine in the synaptic vesicle, waiting to be released again. And so the whole process can start all over again. So acetylcholine esterase breaks down the acetylcholine, so this stops the muscle contraction. The calcium moves back into the sarcoplasmic reticulum by active transport. It can stay present, so it can be used again. The linkage is broken. The tropomyosin blocks the binding site on the actin, so it's like untwisting. Remember, we untwisted the rope, now we're twisting it back, and it's blocked those binding sites on the actin. If the tropomyosin, the troponin, does not have calcium to change the shape of the tropomyosin, then the binding sites will not be visible or exposed to bind with the myosin, so everything is on hold. The myosin and the actin binding cross-bridging is prevented. Now, where do you get energy for contraction? There's two ways to get energy for contraction. And remember, we said that the ATP is the currency for the energy required for muscle contraction. Cellular respiration. Most ATPs come from aerobic respiration. What's aerobic respiration? It is cellular respiration with oxygen. anaerobic respiration is cellular respiration without oxygen. Now, with oxygen, more ATPs are made. So most ATPs are made by cellular respiration, aerobic cellular respiration. And this all happens within the mitochondria. Okay? It is the energy for muscle contraction. A little bit of ATP can be made by anaerobic respiration, and when you are working out real hard, your muscle cannot keep up with the oxygen needed for cellular respiration, so a little bit of ATP can be made by anaerobic respiration, and that provides a little energy for muscle contraction. Now, another component is involved in making ATPs. Now remember, ATP is ADP plus phosphate to form this ATP. The ADP is diphosphate, 2 phosphates. Triphosphate is 3. So you have to add a phosphate for an ADP to become a ATP. And because we're making a larger molecule from a smaller than the energy is stored, ATP is not that helpful for us. It's really not that great unless you break it down from ATP to ADP and release that energy. Okay, so that's what happens for things to work, is the ADP is broken down into ADP plus a phosphate. Okay, so to make this ATP, phosphates are added to ADPs. Now, creatine phosphate. Creatine has a phosphate attached. So when cellular respiration is high, then we can use the ADP from cellular respiration. When cellular respiration is used up, the ADP from cellular respiration, we can also create some ATPs by using creatine phosphate. Now here's this ADP in this cycle, and at a point the creatine releases its phosphate. It is added to the ADP to make ATP. That's called phosphorylation. And so that ADP is phosphorylized, or a phosphate is added to make an ATP. And then you just have the creatine. And then this breaks down into ADP plus phosphate, and then you can make more creatine phosphate. So it's a cycle. Alright? As it's breaking down that creatine phosphate, phosphate is added to the ADP to become ATP. So that's how we get ATPs, the currency for muscle contractions and a lot of other activities, those metabolic activities in your body. Creatine phosphate stores energy that quickly converts ADP to ATP. Okay, so there's stored energy here. It contributes, the energy is added to this ADP as it is phosphorylated. It gains that phosphate and it gains energy, and then the energy is used as this ATP is broken down again into ADP plus phosphate. Okay, again, the aerobic phase makes the most ATPs. Look at all these ATPs. Aerobic means with oxygen. Anaerobic without oxygen. The oxygen, this represents a mitochondria, the cristae of a mitochondria. So the citric acid cycle and cellular respiration occurs in the mitochondria, but it only happens if oxygen is present. Now this is a cell. This is representing a cell here. And this area is where the cytoplasm is. And in the cytoplasm is the jelly-like substance called cytosol. In the cytosol you can have the production of some ATPs, but this does not require oxygen. When we're talking about the aerobic phase occurs in the mitochondria, the powerhouse of the cell. And in the mitochondria, you have the citric acid cycle that produces a couple of ATPs, and then the electron transport chain that produces 32 more ATPs, like 2 in the citric acid cycle and 32 in the electron transport chain, for a total of 34 ATPs. Okay, the citric acid cycle. So you have 36 when you add these. But anyway, the citric acid cycle and the electron transport chain make most of the ATPs with oxygen, the aerobic phase of cellular respiration. In the absence of oxygen, sufficient absences, glycolysis leads to lactic acid accumulation. Okay, so if oxygen is not present, then this pyruvic acid. Now what happens is the breakdown of the ATPs and glucose. Glucose loses some carbons. Glucose, its chemical formula is C6H12O6, and it has a lot of carbons. So it loses some carbons and some hydrogens, and it becomes lactic acid. If it does not have oxygen present, pyruvic acid becomes lactic acid, and you have lactic acid accumulation. If there is oxygen present, then the pyruvic acid goes into the citric acid cycle, turns it twice, and that's how you make all these ATPs. Oxygen is carried from the lungs by the hemoglobin in red blood cells, and it's stored in the muscle. The cells of the myoglobin provide the oxygen and is available to support aerobic respiration. The hemoglobin is the red part of the blood cells. Heme, iron, gives it the red appearance. Myoglobin is what makes muscles that dark brown or dark reddish brown color. Myoglobin, it holds oxygen just like hemoglobin on a red blood cell. But anyway, so the aerobic phase produces most of the ATPs. Now the anaerobic phase again happens in the cytosol, in the cytoplasm, and only a few ATPs are made. But when you are exercising, you get into what is called oxygen debt. And so you cannot produce enough, or you don't have enough oxygen to make a lot of ATPs from aerobic respiration in the mitochondria. So you're going to make a lot here. It just takes more time, and the pyruvic acid, if oxygen's not present to continue down in the citric acid cycle, will be converted to lactic acid. And the lactic acid is stored in the muscles and in the liver, and can be converted back to pyruvic acid and pushed on into the citric acid cycle when oxygen levels are restored. So you've been working out and you're in the cool down phase. You are breathing hard, and then your breathing slows down. Your oxygen levels are being restored. So the lactic acid, if you work out, you know you need to cool down, and that helps to keep moving, helps to move the lactic acid out of the muscle and back into use and into the citric acid cycle with added oxygen. So the anaerobic phase, the prefix "an" means without, or aero is oxygen, so the anaerobic phase is without oxygen, and we call this phase glycolysis. Glycolysis occurs here in the cytosol, not in the mitochondria. So glycolysis is added with the aerobic phase, the citric acid cycle in the electron transport chain to make all the ATPs. Okay, the anaerobic phase makes very little ATPs, and lactic acid is produced if oxygen is not present. Oxygen debt, the amount of oxygen needed by the liver to convert lactic acid into glucose. Now, if lactic acid is converted back into glucose, you get lactic acid from not having oxygen when you're working out, and the lactic acid builds up in the muscle and then is stored in the liver. It can be converted back into glucose when oxygen is present, and then it can be plugged in for the citric acid cycle again and used to make more ATPs, etc., etc. So glycogen is another way to store this pyruvic acid or glucose. If you don't use it all, glucose can be stored as glycogen in the liver, converted back into glucose whenever you need it. Glycogen can be stored in the liver. This is excess glucose. Be stored in the liver and used when it's needed. Now, after it's stored so long, it converts to fat. Okay. So, the glycogen can be made into glucose, and it can provide the energy to synthesize ATP. Synthesize means to make. After it synthesizes the ATP, it is broken down into pyruvic acid. Remember I said it's called decarboxylation. It's losing some carbons and a hydrogen, and it becomes pyruvic acid. And then it loses more to become lactic acid. In glycolysis, there's lactic acid formation in the muscle. Synthesis of glucose from lactic acid happens in the liver so that lactic acid can be converted to glucose. These atoms are added back to it, so it's glucose again, and it will provide energy to make more ATP. This oxygen debt happens when oxygen is not available, when you're working out real hard, or if you're not in good shape. You cannot utilize or deliver enough oxygen, so you will get into oxygen debt. Glycolysis continues. Remember, glycolysis is anaerobic. It's without oxygen, occurs in the cytosol, not the mitochondria. Pyruvic acid is converted to lactic acid because oxygen is not available. The liver converts the lactic acid to glucose. But if you have a whole lot of lactic acid, it can cause a pH problem, and the liver will not be able to convert all of it back to glucose. So lactic acid can be turned back to glucose, and glycogen back to glucose to be used to make more ATP. Muscle fatigue results in the inability of muscles to contract. It's commonly caused by a decrease of blood flow, ion imbalances. Now, we talked about a couple of things that you have to have. You have to have calcium, and calcium is an ion. If you don't have the calcium, then it's going to influence the action of the muscle. But you also have to have sodium and potassium and chlorides. Those ions need to be in check too if you're not taking in enough sodium, which there's not many people who can get into trouble by not taking in enough sodium in their diet. Most of us take in too much sodium. But ion imbalances, the accumulation of lactic acid, can also cause muscle fatigue. And a cramp is a sustained involuntary contraction. It sort of holds tight and doesn't relax. We call it a Charlie horse, especially if you've been walking a lot or running or been standing on your toes for a long period of time. You lay down to rest and all of a sudden you have a Charlie horse. Well, this is a cramp. It's also called a tetanic contraction. Sustained involuntary contraction due to the lack of ATP needed to move the calcium back into the sarcoplasmic reticulum and break the linkage. It can also be caused by too much acetylcholine, the acetylcholine not being broken down. A byproduct of work is heat production. That is, heat energy is given off. It's like a waste product of work. Your muscles contracting, it's doing work, and so you have heat production. It's also a byproduct of cellular respiration. It is the major source. Muscle contractions are the major source of body heat. The blood helps to transport the heat throughout the body, and it helps you maintain a constant body temperature. Now let's just review over, you got this in the lab manual. Let's review over skeletal muscle actions, parts. The origin is the movable end. The origin here, this is the origin of the biceps brachii. Okay, so the coracoid process here, these are parts of the scapula. The coracoid process and above the glenoid cavity on this scapula are the origins of this muscle, and the insertion is the movable part. So the insertion will move a part toward the origin. A prime mover, also called, sometimes referred to as an agonist, is the primary muscle responsible for the movement. Now, this is the biceps, and remember back here would be the triceps. When you are flexing your elbow, now reviewing movements in joints, flexion would bring the this insertion closer to the origin. It would bring this fist closer to the body. This would be flexion, bending that elbow. Okay. Flexion happens as a result of, it is the action of the biceps brachii. Now, so that you don't hit yourself in the face, it needs to be a controlled movement. So the triceps, when you are flexing this forearms, the triceps are actually lengthening at a controlled rate, and the biceps are shortening, okay, contracting. So the prime mover for flexing that elbow would be the biceps brachii. The antagonist resists the prime mover's action and cause movement in the opposite direction. So you have a controlled flexion because you have this lengthening, controlled lengthening of the triceps. So the triceps would be the antagonist. It acts against the prime mover, also called the agonist. Okay, the prime mover has the antagonist to help control. These are working together. Okay, and that's usually called synergy when they work together. Especially in your legs, you have several muscles that are going to help you to bend your leg or flex your leg. Now, extending this arm is called extension, flexion, extension, flexion, extension. So for you to flex, it would be the biceps brachii. To extend this arm going this direction, the triceps brachii would contract, and the triceps brachii would contract and the biceps would lengthen to help control this extension, bringing this hand down. Okay. Now, synergists are muscles that help in a movement. They help the prime mover. Muscles, the action of the muscles that move the body part lie above it. The muscles and bones function together as levers to allow the body movement. And when they work as levers, the joints or the fulcrums or pivots. We talked about that before. The bones are the rigid bars and the muscles provide the force. And we talked about that when we talked about the skeletal system. Okay. Now, how do muscles get their names? Some names are derived from their action. And the abductor longus, it's a long muscle that abducts the leg. Okay? It abducts. Remember, abducts. Brings it closer to the midline of the body. Taking it away would be adduction. This review. Abduction brings it closer to the midline. So the abductor longus is a muscle that brings that leg closer to the midline. Sometimes it's named for the location it's found in, the rectus femoris, right over the femur, rectus femoris. The shape, triangular shape of the deltoid over in the shoulder. Also the trapezius and a shape of a trapezius rectangle. The numbered division, the biceps brachii. Bi means two, the divisions of the head, and seps is a head. Okay, bi means two. The triceps brachii on the underside of the arm has three heads, so it has three attachments, three origins, and one insertion. The biceps brachii has two heads, 2 points of origin with one attachment. Biceps brachii. The point of attachment, the origin is always first, followed by the insertion. Now, this is not one that you had to remember, but the origin is going to be the sternum clitomastoid. So, along the area, the sternocleidomastoid on the bottom side of the jaw and the sternum. Okay, sternocleidomastoid tells exactly the points of attachment of that muscle. The size, magnus and longus, long and big. Now, we're not going to talk a whole lot about smooth muscles here. We'll talk a little bit more about them when we talk about where they're located in other body systems, such as the intestines for digestion and in the walls of the stomach, etc., etc. Compared to skeletal muscles, they're shorter. They have a single nucleus, and remember we said the skeletal muscles have many nuclei in their fibers. A smooth muscle has a single nucleus. They are elongated with tapered ends, so it looks sort of like this. Sort of spindle shape with a bulge in the pointed on both ends with a bulge in the middle where the nucleus is. And sort of long. The myofilaments are randomly organized, not organized like the actin-myosin and the filaments in the skeletal muscles. Not the same. Because they're randomly organized, there are no obvious striations. It lacks transverse tubules to deliver those ions, calcium and all. The sarcoplasmic reticula is not very well developed. Cardiac muscle is found only in the heart. The muscle fibers are joined together by intercalated discs. Intercalated discs, the function of that is to propagate a muscle impulse from one fiber to the next, and also communicate between one fiber to the next. And unlike skeletal muscle, the fibers will branch, so they'll have Y's in them. A network of fibers contracts as a unit. They're self-exciting and rhythmic. Self-exciting means that after the nerve, if you pull the heart out of the body, it may continue to beat because that tissue is self-exciting and rhythmic. It has a longer refractory period than a skeletal muscle. So a refractory period is a time where it receives stimulation perhaps, but it does not contract. So that's a refractory period. It has a longer refractory period than a skeletal muscle. Okay, that is the end of Chapter 8 and the study of muscles. On Blackboard, there are several animations that you need to watch that go along with this lecture. I didn't want you to have to bear watching the animations within this video, so watch them on your own and review and use them to answer your questions.